My name is Bhanwar Lal Puniya and I am going to present my poster on metabolic drug repurposing for autoimmune diseases. I'll be focusing on metabolism of CD4 plus T cells and three autoimmune diseases: rheumatoid arthritis, multiple sclerosis, and primary biliary cholangitis. CD4 plus T cells are essential part of the human immune system. Activated T cells stimulate cytotoxic T cells and natural killer cells to kill infected cells. They can also pass signal to B cells that neutralize the antigen. Metabolism of CD4 plus T cell shapes different CD4 T cell responses. During an immune response, CD4 T cells are activated and proliferate. If we compare the metabolic pathways of naive Th1, Th2, Th17, and T regulatory cells, they differ significantly. The dysregulation in their metabolism may cause impaired and hyperactive immunity, and that is linked with several immune-related disorders. Therefore, CD4 T cell metabolism can be used as a therapeutic target, and a systemic investigation of CD4 T cell can be useful. Thus, we aim to explore CD4 T cell metabolism as a target for autoimmune diseases. First, we focused on characterizing metabolism and developing metabolic models of CD4 plus T cells, and second, to target metabolic genes for autoimmune diseases. Our approach used different steps. In step 1, we collected transcriptomics and proteomics data from public repositories and used them with Recon 3D as a template to construct metabolic models for CD4 T cells. In step 2, we used connectivity map and drug databases to map drug targets on metabolic models. In step 3, we simulated response to drugs or compounds in the model by performing inhibition analysis and finding altered fluxes. In step 4, we analyzed transcriptomics data of patients and identified differentially expressed genes and compared them with flux patterns under gene inhibition. Finally, we used a scoring method and ranked and identified drug targets and repurposable drugs. By integrating transcriptomics and proteomics data, we identified around 6,000 to 9,000 active genes across naive Th1, Th2, and Th17 cells. The model we built using this data and Recon 3D as a reference were consisting of approx 5,000 reactions, 1,000 genes, and 3,000 metabolites. Next, we validated our model with known biological information about T cell metabolism. Here I am showing three specific examples. First, our model showed dependency on glucose. When we reduced flux through glucose uptake reaction, the growth of CD4 T cells was reduced in all the models. Second, lactate production was reduced when flux through pyruvate dehydrogenase was increased. And all the models showed dependency on growth on glutamine, however, under glucose limiting condition. Our model were able to reproduce many other biological behaviors. To use these validated models for drug targets, we identified up and down regulated genes from patients' data, and in the model, when a gene inhibition was simulated, that led us to find up and down regulated fluxes. By comparing up and down regulated disease genes with up and down regulated fluxes under gene inhibition, we identified all the instances where gene inhibition reversed the direction of disease genes. By using this information with perturbation effect score, we ranked and identified drug targets and potentially repurposable drugs and compounds. We identified 17 drug targets for rheumatoid arthritis, 27 for multiple sclerosis, and 24 for primary biliary cholangitis. As an example, we found two drug targets in tyrosine metabolism. One is COMT, which is catechol O methyltransferase, and other is ADH5, which is alcohol dehydrogenase 5. Next, we experimentally validated some drug targets. I'm showing one example in which we use drug entacapone that target COMT at 100 micromolar and 1000 micromolar concentrations, we found that CD4 T cell proliferation was significantly reduced at two time points, 48 hours and 72 hours post drug treatment. In summary, we developed metabolic models of naive Th1, Th2, and Th17 subtypes. We used a novel scoring method. We identified 68 drug targets, and approximately 50% of these targets were validated with either in-house experiments or with existing literature to show the inhibitory effect on T cell proliferation. Also, we used our approach and created a pipeline for metabolic drug target identification and drug repurposing that is implemented in Python and available as a Docker image. Finally, I'd like to thank all the co-authors contributed to this work and funding sources NIH and Systems Biology grant of the University of Surrey. Thank you.